Hi, today I'll be discussing with you a very important publication that I found interesting, and the title of the work was RoseTTAFold Diffusion Guided Short Peptide Design, a case study of binders against KEEP1 NRF2 complex. So the publication was released early this year, 2025, in the Journal of Computational and Structural Biotechnology. So the reference publication is indicated even in the slide. So my name is Fatoki. So today I'll be walking you through this particular publication, and I believe that you will gain one or two things even in this presentation. Okay, so the overview of the study. So the objective of the study was to design peptides that target KEEP1, which is a very important protein that regulates a lot of signaling pathways within the human body or in some most of the organism. So this particular KEEP1 is a central regulator of KEEP1 NRF complex pathway. So in this publication, the specific objective were in threefold. Number one, they want to design short peptide sequence using RF diffusion, that is RoseTTAFold diffusion, and whatever result that they obtain from that, they optimize it using protein MPNN, which is a kind of advanced neural network. Then objective number two was to determine critical properties such as the toxicity of the peptide that they designed, the allergenicity of those peptides, then the aggregation propensity and their solubility. So this will help them even to predict the pharmacological profile of each of the peptides to find them either suitable or unsuitable. The objective three was to understand the interaction and the stability of a KEEP1 peptide complex by using molecular dynamics simulation. So the overview of the work in the paper were presented even in this particular way, where you have even the backbone design even of the peptide using RF diffusion, and then the protein MLP was used even to generate even the actual sequence that can bind even to KEEP1, and then they using silico analysis to identify different peptides that are biologically relevant, and then molecular dynamics simulation was conducted to understand even the overall stability of the interaction of the peptide KEEP1 interaction. So, but we need to understand the biology even of this process, which is called the protein-protein interaction, PPI. So PPI play a vital important role in numerous physiological and also pathological processes such as cancer, neurological neurodegeneration, and also in viral infection, among others. In normal circumstances, normal physiological states, protein-protein interaction occurs in order to bring about signaling of a normal homeostasis of the cell. But also in the case of a disease, for example, cancer, so the aberration of this protein-protein interaction could favor the what's called proliferation of the cancer cell and the degeneration of the neurons in the case of neurodegenerative diseases. So we have a lot of examples even of PPI, but there are some that has been specifically implicated even in cancer. So I listed even just about 8 here, and the last one, KEEP1 NRF interaction, was the focus even of this paper that I am presenting even to you even today. So in order for us to understand, okay, what are the inhibitors that can stop the interaction of KEEP1 NRF or interaction of protein-protein interaction. So we have a study have shown we have two major kind of inhibitor. You can see even on the screen. we have the orthosteric inhibitor, that is the one that binds to the active sites of one of the protein and then prevent others even from binding even to that site even on the other protein. Then we have another inhibitor which is called allosteric inhibitor. So this one doesn't bind even to the catalytic site; it binds to the another binding site even on one of the protein, and this binding to the other side even on the protein affects the catalytic pocket even of that particular protein in such a way that that protein will not be able to recognize even the other protein that binds even to it. So as a result of that, there is a shift even in the actual catalytic site even of one of the proteins, so that the interaction doesn't bring about any favorable outcome. So we have a lot of PPI implicated in cancer, such as MNM2P53 interaction, uh, C-MIC, MAX interaction, and a lot of them like that. But today, the focus of the paper was to keep one NRF interaction. Okay, so talking about how this happens, so we have a lot of schematics that define what happened even between KIP1 and NRF2, and how this condition, which happens within the cytoplasm, upon the NRF2 uh, movement even into the nucleus, then there is a lot of induction of different genes even within the nucleus, which affect even the processes of antioxidant response. Okay, so keep one not only binds even to NRF2, you can see it can also bind even to P52, P62, okay, which leads to protosomal degradation. And also you can see that NRF that got even into the nucleus can also bind to another protein called NAF, and that one even is the key major target that the recent study is being focused on in order to understand what really happens even within the nucleus, Okay, so study is being going on, okay, to prevent, okay, the keep one NRF that occur even in the cytoplasm. Okay, how does that interaction, how does that interaction favors even cancer or not? So there is a debate ongoing even in that on that particular interaction of NRF2 KIP1. Okay, so some are saying, okay, it's not really effective. That what happens even within the nucleus is the most important thing that drives even the proliferation of genes that are related even to cancer. Okay, so you have the references over there. So the redox activation of a transition factor, which is NRFE2 related factor 2, that is the full meaning of NRF2. Okay, so that redox activation is the key regulator of cellular defense, both against the extrinsic and intrinsic oxidative and electrophilic stress. We call it oxidative stress. Okay, so when there is oxidative stress, then there will be activation of NRF2. Okay, then keep one also, which regulates the activity of NRF2. Okay, once it binds to the N-terminal sequence of NRF2, then it will what? It will it will regulate the affairs of NRF2. Okay, it binds to the N-terminal sequence of NRF2 to regulate the affairs of NRF2. But NRF2 in cancer is activated through different mechanisms. We have a lot of about 3 mechanisms by which NRF2 could be activated, not just even through KIP1. Okay, so but the most important or the most common one is through KIP1. Okay, so a common mechanism of NRF2 activation is through the gain of function, GOF mutation of NFE2LE, which is a protein, okay, that also regulates even NRF2. And also we have the loss of function mutation in KIP1. Okay, if there is mutation in KIP1 that, okay, is unable to regulate NRF2, and then the level of NRF2 becomes increased, okay, that will drive and leads to a lot of gene expression within the nucleus and all those things that happen even in cancer. Okay. So in the paper that I'm presenting to you today, so the approach that they used was that okay, they want to design peptides that has a residue, amino acid residue of 3 to 10, and then which can bind to KEAP1 protein. KEAP1 protein is large than NRF2. NRF2. Okay, so they focus on the large protein within the PPI. Okay, that is the protein-protein interaction. So they focus on the protein that really actually has the binding site, the catalytic site. Okay, so they focus on that particular protein KEAP1. So they use RF diffusion to generate models of peptide, that is the backbone of peptide that can fit into the pocket even of a KEAP1 protein. So they use the PDB of a KEAP1 to FLU, the PDB ID of a KEAP1. So RF diffusion, RoseTTAFold, if you heard about RoseTTAFold, that is the basis, but it builds even on RoseTTAFold in such a way that it integrates a lot of internal models in order to predict the backbone of protein that can bind even to the given catalytic sites. So these are the overview of what diffusion model is all about. Okay, so if you look at it, we have the generation step. Okay, so it actually starts by suggesting even a particular sequence, that is the RoseTTAFold. Okay, so the RoseTTAFold, if you look at it, we have input of sequence, we have homologous templates, and then we have prediction of structure. But in RF diffusion, so it's obtained even from the given coordinates. Okay, so it generates even the sequence that can fit in even to that coordinate. So RF diffusion doesn't need what's it called now, homologous templates. That is, there is no need of homology modeling even in RF diffusion. So it basically use a kind of diffusion of the coordinates. That is, you rearrange and permutate even the coordinates based on the given pocket, and then sometimes you need to specify even the actual residues that are very important even in that particular pocket. Okay, so they design specific peptide sequence. Okay, so the design specific peptide sequence, okay, was optimized. Okay, the backbone sequence that was obtained from the RF diffusion, okay, were optimized using protein MPNN, which is a kind of a deep learning approach that combine AlphaFold for structure prediction. Okay, so RF diffusion generates even the what, then give you the backbone. Okay, then protein MN help you to generate the sequence, and then it was integrated to the AlphaFold. So as it generates the sequence, then it's used AlphaFold to build even the structure. So it was so automated using protein MMP. So automated the process, you obtain the sequence, and at the same time it integrates it even to AlphaFold, and then it obtain even the structure. So the structure actually fits even into the pocket of KEAP1. So this approach doesn't require that you conduct molecular docking, because AlphaFold generates the structure immediately based on the binding post that this sequence form even on the pocket of KEAP1. So MPNN means message passing neural network. Okay, so it operates using a kind of undirected graphs, as I've said, with nodes, node feature and edges. Okay, so it's randomized even the sequence, and then find out of the 20 amino acids, essential amino acids, find the amino acid sequence that fits the backbone generated by the RF diffusion, and then that match even the catalytic pocket of the protein that is under the study. So these are the overview of how MMP even work, and then the GitHub link, I provide it even in this particular slide. Okay, So, and that is how the sequence was converted even to the structure, and then the structure bind even to the target, and then the complex structure now with the output, that is, will be the output even of this particular process. So you have the structure already bound even to your protein of interest, that is the key point, the peptide that has been designed. The structure has been generated in such a way that the post is what is on the key point structure. That is, so you have the complex structure as the output even of this particular process. So the GTOP link even to this code is also provided even in this slide. So, once they generated even the sequence, and then they generated the structure and then the complex, okay, so they want to find out, okay, it's not just about peptide binds even to the pockets. Does it really have the biological what's it called now, stability or biological relevance? So that was what led them even to conduct in silico analysis of the identified peptide. So they use arrays of in silico methods to screen even the peptide that they generated even in the study, in order to find those set of peptides that are biologically relevant. So they use a lot of toxicity prediction, allergenicity prediction. So also they use this particular database, UDLMP prep. Okay, so that one, okay, help them to determine activity of the peptide in terms of antioxidants activity, because if you are dealing with cancer, so antioxidant activity of a peptide is very important. If you are considering other study, let's say you want to study something related to inflammation, then anti-inflammatory activity, or that will be the activity that should be tested. So based on the study, based on the objective, which is cancer, okay, key point is related to cancer. So that's why they test for the activity that are related to antioxidants. Then they use a lot of other predictive tools in order for them to get the physiochemical property of each of the peptide in terms of the half-life, the solubility, and then the propensity even for that particular peptide even to penetrate the membrane, okay, to penetrate the membrane, because cell-penetrating peptide prediction. Okay, so after they have been able to screen all these peptides, they generated a lot of them. So they find some that are biologically relevant, and then they subjected those biologically relevant peptides into molecular dynamics simulation in order to understand the stability, conformation state of these peptides. Okay, so they conducted the molecular dynamics simulation using Maestro 2023-2024 suite, which was implemented in Desmond, okay, from Strundinger. Okay, so they use a normal protocol. That is, they use the T3P water model. Then they add the salt NaCl even to adjust even to neutralize even the system, and then the system was subjected to energy minimization and then equilibration using MPT ensemble at 1 atmosphere and then 300 Kelvin. And then the whole actual MD production was conducted at for a kind of 100 nanoseconds, using OPLS 4 phosphate. So what were the results of this particular study? So RF diffusion generative model produced 1000 biodal short peptide backbone. Okay, it says short peptide within 3 to 10 amino acid residues. So RF diffusion was able to generate 1000. So it was specified. Okay, in the model you can specify the number of output that you want, and then you define the hotspot of the catalytic side of your protein. So that is on keep one. So they specify, okay, this amino acid 502, okay, amino acid 550. So these are the key hotspot amino acid that define even the catalytic site. Okay, so RF diffusion generated peptides that fit even into the pockets, generated peptide backbone that fit into the pockets. Okay, so Now, when they use a protein MPN, okay, so they did it even in three sequence of in three generative mode of 189 peptides. Okay, so PLNP was able to, out of the 1,000 backbone, was able to produce 5, 6, 7 peptide sequence, 5, 6, 7 peptide sequence. Okay, so the graphical output even of the work are shown even on the screen. Okay, so out of the 5, 6, 7 peptide sequence that were generated, okay, so the in silico analysis were conducted even on this uh, number of peptide sequence, that is 5, 6, 7 peptide sequence. But out of all the 5, 6, 7 peptide sequence, only 11 peptides were found to be novel and also pass the in silico assessment. That is, out of 5, 6, 7, only 11 were found to be biologically relevant, pass through all the screening, antioxidant screening, allergenicity, and all the stuff like that. Okay, so 8 were found to have a good solubility. That is, they will favor oral routes. Okay, and then only 2 are found to have poor stability, but they pass through all the other, they pass all the other assessments, but they were not soluble. So that is not bad. So they can still undergo a kind of optimization or pass through subjected to different route of administration. Okay, so the data about the other physicochemical properties of this 11 peptide were indicated even in the table as being shown even on the slide. So we have the solubility, cell-penetrating capacity, then we have the half-life of each of the peptides, and then we have the novelty. We have the novelty even of how the peptide is it in existence or is it a new peptide entirely. Okay. So these 11 that passed through even in silico analysis were taken further even for other soil analysis. Okay. So out of the 11, 8 ones that has a good solubility were subjected even to molecular dynamic simulation. Okay, so the standards, which is the NRF2, was also even subjected to molecular dynamic simulation. Okay, so the results that they obtained, that is, they analyzed the trajectory, the output of the MD simulation. So they analyzed the trajectory for RMSD, root mean square deviation, RMSF, root mean square fluctuation. Then they analyze the radius of gyration, the SASA, surface accessibility, surface area, then the number of hydrogen bonds, and they conducted a kind of a molecular mechanism to calculate the binding free energy of each of these peptide. Okay, if you talk about RMSD, that is, they want to check even the deviation even of the structure from the initial structure. Okay, from the initial structure, is the structure stable over the period of the simulation? The RMSF, they want to check the atomic fluctuation of each of the residue in form of their position. Okay, is the position shifts during the course of the simulation? Then the radius of gyration, the whole structure, to what extent does it gyrate during the period of simulation? That is a kind of translational or the rotational movement even of the structure of the peptide away even from the initial position. Then the SASA, okay, this particular peptide, do they have in form of the interaction even with the environment, with the aqueous, that is the water that is being simulated with? How does the amino acid residue, how does they post themselves? Okay, is it that, okay, hydrophobicity side cluster in one side while the hydrophilicity side even cluster in one? So these help us even to predict how the peptide behave even in the physiological situation or solution. Okay, so they find out that this particular peptide, that is the one with PNPIVE, has the overall stability in form of a RMSD. It has the lowest RMSD, that is, the structure does not fluctuate. And then also you can see all the RMSF, they are within this almost the same range. And then the RG also almost the same range. Every other parameters remains almost the same, apart from that RMSD where PIVE peptide, that is peptide 431, has the lowest deviation in terms of the structure. But this particular publication showed us a kind of standard deviation, that is, showed us a kind of statistic in form of a plus or minus, but in the publication, they did not show us the number of times the simulations were conducted. Okay, that's why I put there n is equals to unknown. Is it 3 times? Is it 5 times? They repeated even this particular simulation. So the publication did not report that, which is not good. So part of the sampling that was done even from the MD is what I show even on the slide. So this particular structure was reported even in the as part of the supplementary publication. Okay, so also the net charge even of each of the peptides. No, charge is very, very important even in terms of membrane interaction. So looking at NRF2, the net charge on NRF2 is -4. Okay, so peptide 93 also had -4, and then the interactions also, that is the interaction site even on the KEEP1 was similar even to that of NRF2. Looking at that peptide 431 PNPIV peptide, it has a -1. So that is a kind of a signal that it will have a kind of a limitation when interacting with the membrane. However, it has all the same binding interaction, even like that one of NRF2 also. But looking at the free energy MMGBSA results, so we can see even from here that when NRF2 has minus 89.47 kilocalorie per mole free energy, while the peptide 41 has minus 76. It's very good. So I think it has the next lowest free energy like NRF2. So overall, this particular work favors even this peptide 431 with the sequence PNPIVE. So they reported the free energy landscape of KEEP1 peptide complex, where they plotted RMSD against the RG. But this kind of plot does not necessarily give us any particular information about what is going on, about even the what's going on even within the complex, because RMSD is not it's not reliable. It's not reliable. So this kind of plot does give us a pictorial of the interaction of what's going on during the MD simulation, but not necessarily giving us specific information about the complex. So what are the pros and cons that I found even in this particular work? Though I gave all the references even to what I put up over here. So you can see that the pros, they integrated AI, that is using RF diffusion, which is a new model of peptide design. So they integrated this with the in silico analysis, and then they conducted the molecular dynamics simulation. Then number two, in silico analysis, especially the enzymatic cleavage prediction, because they predicted, okay, what are the effects of trypsin, chymotrypsin, and these processes that are present even within the intestine or stomach. So they simulated that to find out which one we escape the hydrolysis even of by these enzymes. So it helps to filter from 5, 6, 7 peptides. It filter this equalizer helps to filter it even to 8 peptides that are biologically useful. But if you consider the cons, that is, the negative part even of this work is that it was computationally intensive. Okay, so if you want to repeat that, then you need a lot of resources even to implement that. So they use a PC desktop computer that it has that is Core i9 processor, and then they have this GPU facility, NVIDIA RTX 4090, which is a very powerful one. And then the size of the RAM was 200 gigabytes. So this is a kind of a customized computer. So customized computer. So looking at the price to build this kind of a computer is because approximately about 7,000 US dollar. Okay, so I try to check even the link to similar equipment, similar computer even online. Okay, so you can see, but the study is very good, but it is computationally intensive, and then to replicate it, you need a lot of facilities and support even to repeat this kind of process. Then currently, the indicia of KIPO NRF2 is contestable, just like I discussed even at the beginning even of this particular work, that NRF2 KIP1 complex happens within the cytoplasm. So it is when KIP1 is unable to regulate NRF2 and then SSNRF2 moves to the nucleus. So within the nucleus, there is another binders to NRF2, which is MAF gene. Okay, so MAF gene is now being targeted as even the more important even than KIP1. Okay, there's a publication that supports that, where they try to embed interaction between NRF2 and the MAF gene. Okay, that is Modi et al. 2023. You can read that and explore more about this topic. So my recommendation, yes, the work was so good. You use the latest AI model, RF diffusion, but what has the overall outcome even of the work? The overall outcome gave us just 8 peptides, despite all the input, energy, expense, resources wise, intensive wise. So you can see that the whole work doesn't even really worth it. And yes, it is possible to incorporate enzyme hydrolysis learning module into the design pipeline. That is, if there is a way to incorporate enzyme hydrolysis into the RF diffusion, okay, that will go a long way. That will go a long way because it will be a critical step for cost-effective computational discovery of a biologically useful peptide. Okay, so what is the essence that we generate 1,000 peptides using very sophisticated equipment, resources, and then at the end it's only 8 that were found to be biologically relevant. So it's better to turn the situation around, where you design algorithm that incorporates enzyme hydrolysis, that is, effect of processes on this particular peptide into the model, so that you know that all the expenses that you have been all the expenses will give you very important, very biologically relevant outcome. Okay, so enzyme cleavage sites, for those that want to write the code, you can follow this particular spacy.org peptide cutter, and then you follow the instruction over there. You see different sites where each of the peptide works. Okay, so this kind of work is an ongoing area where you want to design a peptide that can bind to a particular complex or a particular single protein. So there is this particular complex that this particular company, Adaptive Bio, always run every year, where they bring together different computational experts, where they try to design a binder for their given protein, especially the EGFR protein. Okay, so they will rank and use their AI model to screen those peptide that binds even to this particular protein. So the last one that was conducted, so 1,000 binders were submitted, but about 30 were found to be biologically useful in vitro. In vitro, okay? So, but out of those 30, we don't know how many will be what we finally have boostability in the eventually. So we can see even from here that yes, AI model and everything is good, but you must understand the biology. Okay, you must understand the biology and know how to use your resources very well. Know how to use your resources very well. Okay, so guys, this is what I have for you even today. So you can access the publication. You can access the publication even online, and then review the supplementary material, and then improve your skill. Learn about RF diffusion. Learn about protein MPLA. Learn about in silico analysis. Follow all the links that were provided. Learn about molecular dynamics simulation. Also understand the biology, enzyme proteolysis, okay, trypsin, chymotrypsin, how do they affect even the peptide, okay, and all these, you put them together into your biological, into your computational understanding. It will help you in designing a better study and also to impart even the ongoing to provide solution even to the existing problem facing us even as human being. Okay, so in my upcoming talk, I will try to inform you about how to use algorithm that puts into consideration enzyme proteolysis effects into your design even of peptide, to your design of peptide. Okay, so that is very, very important. And also another way to look at is in the design of all the peptides, okay? That is, all your peptide consists of a D amino acid. D amino acid. That's what I'm conducting my doctoral research even upon. I'm studying all the peptide of a particular antimicrobial peptide, then comparing the effect or the outcome even with that one of all L peptide, Okay, so yes, L peptide is all L peptide is prone to all these proteolytic cleavage. The D peptide has been shown to have a kind of a stability, and also beyond that, there may be possibility even of a chiral effect in the mechanism of action of how the peptide works. Thank you. See you in my next talk. Bye.